Today we see nature of enzymes, classification, purification and kinetic assays, immobilized enzymes and their uses. The topic has been divided into two parts. So, hereby in first part, we will be learning nature of enzymes, purification and immobilized enzymes and their uses. Introduction. The living cell is a site of tremendous biochemical activity called metabolism. This is the process of chemical and physical change which goes on continually in the living organism. Build-up of new tissue, replacement of old tissue, conversion of food to energy, disposal of waste materials, reproduction, all the activities that we characterized as life. Building up and tearing down takes place in the face of an apparent paradox. The greatest majority of these biochemical reactions does not take place spontaneously. The phenomenon of catalysis makes possible biochemical reactions necessary for all life processes. Catalysis is defined as the acceleration of a chemical reaction by some substance which itself undergoes no permanent chemical change. The catalyst of biochemical reactions are enzymes and are responsible for bringing about almost all of the chemical reactions in living organisms. Chemical nature of enzymes. All known enzymes are proteins. They are high molecular weight compounds made up of chains of amino acids linked together by peptide bonds. Enzymes are proteins made up of amino acids. Active sites are formed by secondary and tertiary clustering structure of amino acids, which results in the formation of a physical groove where the substrate binds. Changes on the amino acids allow them to interact with substrate by donating or accepting electrons or protons depending on the enzyme. Enzyme substrate binding involves temporary hydrogen ion and or ionic bond at the active site. Enzymes can be denatured and precipitated with salts, solvents and other reagents. They have molecular weights ranging from 10,000 to 2 lakh. Simple enzyme. It is made up of only protein molecules, not bound to any non-proteins. Example, pancreatic ribonuclease. Many enzymes require the presence of other compounds, that is cofactors, before their catalytic activity can be exerted. This entire active complex is referred to as a holoenzyme, that is apoenzyme, that is the protein portion plus the cofactor, that is the coenzyme, prosthetic group or metal ion activator. According to Holm, the cofactor may be coenzyme. It is a non-protein organic substance which is thermostable and loosely attached to the protein part. Prosthetic group. If the cofactor is bound so tightly to the apoenzyme, it is difficult to remove without damaging the enzyme. It is sometimes called a prosthetic group. It is an organic substance which is thermostable and firmly attached to the protein or apoenzyme portion. Metal ion activators. It includes ions like K positive, Fe2 positive, Fe3 positive, Cu2 positive, CO2 positive, Zn2 positive, Mn2 positive, Mg2 positive, Ca2 positive and MO3 positive. Coenzymes. Coenzymes are derivatives of vitamins without which the enzyme cannot exhibit any reaction. One molecule of coenzyme is able to convert a large number of substrate molecule with the help of enzyme. Coenzyme accept a particular group removed from the substrate or donates a particular group to the substrate. Coenzymes are called co-substrate because the changes that takes place in substrates are complementary to the changes in coenzymes. The coenzyme may participate in forming an intermediate enzyme substrate complex. Example, NAD, FAD, coenzyme A. Metal ions activators. Many enzymes require metal ions like Ca2 positive, K positive, Mg2 positive, Fe2 positive, Cu2 positive, Zn2 positive, Mn2 positive and CO2 positive for their activity. Metal activated enzymes form only loose and easily dissociable complex with the metal and easily release the metal without denaturation. Metalloenzymes hold the metal tightly on the molecule and do not release it even during extensive purification. Metal ions promote enzyme action by maintaining or producing the active structural conformation of enzyme, for an example glutamine synthase, promoting the formation of enzyme substrate complex, for an example enolase and carboxypeptidase A, acting as electron donors or acceptors, example Fe, S proteins and cytochromes, causing distortion in the substrate or the enzyme, example phosphotransferases. Zymogens, inactive form of enzymes. Some enzymes are produced in nature in an inactive form which can be activated when they are required. Such type of enzymes are called zymogens, that is proenzymes. Many of the digestive enzymes and enzymes concerned with blood coagulation are in this group. Examples, pepsinogen. This zymogen is from gastric juice. When required, pepsinogen converts to pepsin. Trypsinogen. This zymogen is found in the pancreatic juice and when it is required gets converted to trypsin. The activation is brought about by specific ions or by other enzymes that are proteolytic. Zymogen forms of enzymes a protective mechanism to prevent autodigestion of tissue producing the digestive enzymes and to prevent intravascular coagulation of blood. Isoenzymes, that is isozymes. Isoenzymes are physically distinct forms of the same enzyme activity. These are enzymes having similar catalytic activity, acts on the same substrate and produces the same product but originated at different sites and exhibiting different physical and chemical characteristics such as electrophoretic mobilities, amino acid composition and immunological behavior. Higher organisms have several physically distinct versions of a given enzyme, each of which catalyzes the same reaction. Isoenzymes arise through gene duplication and exhibit differences in properties such as sensitivity to particular regulatory factors or substrate affinity that adapts them to specific tissues or circumstances. Some isoenzymes enhance survival by providing a backup copy of an essential enzyme. Isoenzymes are expressed only in specific cell types. The expression of isoenzymes in specific cells occurs during certain periods of development or in response to specific physiologic or pathophysiologic changes. Thus, analysis of the presence and distribution of enzymes and isoenzymes are often useful in diagnosis. Isoforms of lactate dehydrogenase is useful in diagnosis of myocardial infarction, while study of alkaline phosphatase isoforms are helpful in diagnosis of various bone disorders and obstructive liver diseases. Example: LDH, that is lactate dehydrogenase, exists in 5 different forms, each having 4 polypeptide chains. H stands for heart and M stands for muscle. Types of polypeptide chain: LDH1, HHHH, LDH2, HHHM, LDH3, HHMM, LDH4, HMMM, LDH5, MMMM. Example: CPK, that is creatinine phosphokinase, exists in 3 different forms, each having 2 polypeptide chains. Characteristic subunits are B stands for brain and M stands for muscle. Types of polypeptide chain: CPK1 BB, CPK2 MB, CPK3 M. Enzyme purification. Enzymes are manufactured in bioreactors for commercial use. These enzymes are in the crude form and have to be purified for further use. The extraction methods are followed by the purification processes. There are mainly 3 major purification methods depending on the technique or property of enzyme. Number 1, based on ionic properties of enzymes. Number 2, based on the ability to get adsorbed. Number 3, based on differences in size of molecules. Number 1, techniques depending on the ionic properties of enzymes. In that, salting out, it is done by varying the pH of the solution or by addition of chemical agents which carry out precipitation. At the isoelectric pH, proteins have minimum solubility and hence get precipitated out as pure crystals. For example, the digestive enzyme pepsin is purified by this process. Salting out is also possible by addition of chemicals such as ammonium sulfate, acetone, etc. Normally used ions based on the decreasing order of ability to precipitate is given below. Anions: citrate, tartarate, sulfate, acetate, chloride, etc. Cations: TH4 positive, Al3 positive, Ba2 positive, Sr2 positive, Ca2 positive, Mg2 positive, NH4 positive. The series is also known as Hofmeister series. As the chemical agent gets dissolved in the aqueous phase, salting in takes place. Proteins get soluble during this phase. Once reaching a critical limit, the protein-protein interaction overcomes protein-water interaction as the agent displaces the water molecules. This results in salting out or precipitation of proteins or enzymes. B. Electrophoresis. It is the movement of charged particles under the influence of an electric field. The ions migrate based on electric charge and strength of the field. Number C, ion exchange chromatography. Selective adsorption and exchange of ions takes place in the adsorption sites of ion exchange columns filled with ion exchange resins. Example, DEAE, that is diethyl amino ethyl. Cellulose is an anion exchanger, and DM, that is dimethyl cellulose, is a cation exchanger. Number 2, techniques depending on the adsorbing properties of enzymes. In that, A, adsorption chromatography. It is based on the principle of adsorption. Solute or enzyme get adsorbed onto the particular sites of the adsorption chromatographic column depending on the effective distribution coefficient. These are then eluted using various solvents. The packing materials used include starch. Effective distribution coefficient is the ratio of distribution of solute across the different phases of chromatography. The process is widely used for initial recovery of extracellular enzymes manufactured on large scale. B, affinity chromatography. This technique makes use of enzyme substrate interaction. A matrix with a ligand is packed in the column. As the enzyme solution passes through the column, the solute or enzyme molecules get attached to the ligand. These are then eluted by suitable solvents. In effect, the matrix ligand complexes remain adsorbed to the column till it gets eluted. Number 3, techniques depending on the size of enzymes. In that, A, molecular sieve or gel filtration or gel permeation chromatography. This makes use of gel material as the supporting matrix. Smaller particles get entrapped in the pores, whereas large particles get through the interfacial space. Hence, the larger particles get separated first, followed by the smaller particles. B, ultrafiltration, that is dialysis. It is the process of passage of solvent molecules from a region of high concentration to a region of lower concentration through a semi-permeable membrane. The technique is used for purification of crude enzymes. It is used for enzyme concentration as well as enzyme purification. Enzymes, when present in aqueous solutions, cannot be retained in the bioreactor. They can be retained in the system by the process of immobilization. It is the process of attaching a cell or enzyme to an insoluble inert support. The purity of such immobilized enzymes is higher than free enzymes. The turnover number is high and enzyme activity can be retained for a longer duration. Other sophisticated methods such as electrodialysis, electrofiltration, isoelectric focusing, force flow electrophoresis and electrodecantation are also developed and used. Membrane based processes such as reverse osmosis and pervaporization are also gaining popularity. For membrane processes, retained species tend to concentrate on the upstream of the membrane and reduce the permeation flux in a process called concentration polarization. Immobilized enzymes. Enzymes display great specificity and are not permanently modified by their participation in reactions. Since they are not charged, during the reaction, it is cost effective to use them more than once. However, if the enzymes are in solution with the reactants and or products, it is difficult to separate them. Therefore, if they can be attached to the reactor in some way, they can be used again after the products have been removed. The term immobilized means unable to move or stationary, and that is exactly what an immobilized enzyme is. An enzyme that is physically attached to a solid support over which a substrate is passed and converted to a product. There are 4 different ways by which one can immobilize an enzyme, which are the following, listed in order of effectiveness. Affinity tag binding. Enzymes may be immobilized to a surface, for example, in a porous material using non-covalent or covalent protein tags. This technology has been established for protein purification purposes. This technique is the only one which can be regarded as generally applicable and can be performed without prior enzyme purification with a pure preparation as the result. Adsorption on glass, alginate beads or matrix. Enzyme is attached to the outside of an inert material. In general, this method is the slowest among those listed here. As adsorption is not a chemical reaction, the active site of the immobilized enzyme may be blocked by the matrix or beads, greatly reducing the activity of the enzyme. Entrapment. The enzyme is trapped in insoluble beads or microspheres such as calcium alginate beads. However, this insoluble substances hinders the arrival of the substrate and the exit of the products. Cross linkage. Enzyme molecules are covalently bonded to each other to create a matrix consisting of almost only enzyme. The reaction ensures that the binding site does not cover the enzyme's active site. The activity of the enzyme is only affected by immobility. However, the inflexibility of the covalent bonds precludes the self-healing properties exhibited by chemoadsorbed self-assembled monolayers. Use of a spacer molecule like polyethylene glycol helps reduce the steric hindrance by the substrate in this case. Uses of immobilized enzymes. Use of immobilized enzymes in different fields such as in medicine, antibiotic production, drug metabolism, food industry, biodiesel production, bioremediation, etc. The development and attributes of several established and emerging industrial applications for immobilized enzymes, including high fructose corn syrup production, pectin hydrolysis, biodiesel production and carbon dioxide. The first reported application is the production of L-amino acids from immobilized amino acylase. The production of a chiral molecule is of principal importance for pharmaceutical and research purposes because isomers formed differ significantly with respect to their biological properties. Use of immobilized glucose isomerase in food industry for the production of high fructose syrup, as fructose is 16th time sweeter than glucose. Immobilized enzymes are used in the production of lactose-free milk for people who are lactose intolerant. Immobilized enzymes are used in glucose testing strips to measure glucose levels for diabetics. A number of immobilized enzymes are also becoming an important part of a variety of biosensors. The term biosensor is used for analytical tools or systems which make use of an immobilized biochemical material along with a suitable transducer device to convert a biochemical signal to a quantifiable chemical signal. Immobilized enzymes have an increasing number of analytical applications, especially in clinical situations where they differ in the potential for fast, sensitive and accurate determination of analytes such as blood glucose and urea. Most significant development is the combination of immobilized enzymes with high specificity and sensitivity electroanalytical chemistry. The resulting enzyme electrode offer an advantage of accurate analysis without the need of sample preparation. Production of antibiotics. The antibiotic penicillin can be produced by using an immobilized enzyme from the culture of penicillin production cultures in fermentation. Immobilized cell and enzyme cultures can be produced other antibiotics like ampicillin, amoxicillin, etc. Application in commercial production. Some of the important immobilized enzymes are amino acylase, amylase, invertase, aspartase, fumarase, etc. Amino acylase is used for production of L-amino acids from D-L-acyl amino acids. Amylase is used for production of glucose from starch. Invertase is used for splitting of sucrose to fructose and glucose. Aspartase is used for production of aspartic acid from fumaric acid. Fumarase is used for synthesis of malic acid from fumaric acid. Another recent development involves the quantification of enzymes, substrates and another analytes in samples of clinical importance. For this, impregnate the reagents including the enzyme onto an immobilized structure. On addition of a drop of test sample to the matrix, the analyte in the sample triggers an enzyme reaction resulting in the formation of colored product. The intensity of the color can be measured with the help of reflectance spectroscopy. Summary: the enzymes are biological catalysts necessary in biological reactions. These are proteins, each having a specific substrate. Many enzymes require the presence of other compounds like cofactors before their catalytic activity can be exerted. This entire active complex is referred to as the holoenzyme. Enzymes are manufactured in bioreactors for commercial use. These enzymes are in the crude form and have to be purified for further use. There are also enzymes called as immobilized enzymes that are physically attached to a solid support over which a substrate is passed and converted to product. They are used in different fields such as in medicine, antibiotic production, drug metabolism, food industry, biodiesel production and bioremediation. In part 2, we will be learning classification of enzymes and enzyme kinetics. Thank you.